Hello and welcome. Dear friends, today we will talk about the mechanism and role of senescence in plants. First of all, let me give you a general account of senescence. The term senescence has been derived from the Latin word senescere, which means to grow old. In general context, senescence means death. The death of cells, organs, or individual plants appears to be programmed and in some sense adaptive. This is clearly so with the death of individual cells during differentiation, when residual products contribute to the effective function of the entire plant body. The death of leaves and of shoot system is part of the plant's adaptation to the cycle of seasons. About a century ago, the word senescence was conceived of in scientific texts as the end phase of differentiation, the phase that culminates in death. Senescence comprises those processes that follow physiological maturity which lead to the death of a whole plant, organ, or tissue at macroscopic level. However, at microscopic level, the process is continuous since there exists always a turnover of cell organelles at one or the other places of the whole body. Senescence is an integral part of the normal developmental cycle of plants and can occur at cell, tissue, and organ or organization level. It's the final event in the life of many plant tissues and is a highly regulated process that involves structural, biochemical, and molecular changes that in many cases bear the hallmark of programmed cell death. Senescence, though a terminal developmental stage, can be accelerated by an array of both biotic and abiotic factors such as light, temperature, nutrients, ethylene, abscisic acid, pathogens, and pollination, etc. It's a dynamic and closely regulated developmental process which involves highly coordinated changes in gene expression and requires active gene transcription and protein translation. A genetically controlled senescence program allows for the ordered degradation of organelles and macromolecules with the remobilization of essential nutrients. It's largely an oxidative process involving a general degradation of cellular structures and mobilization of the products of degradation to other parts of the plants or organs. Senescence is mainly characterized by many changes including cessation of photosynthesis, disintegration of organelle structure, intense loss of chlorophyll and proteins, upregulation of tonoplast localized cytochromes, a dramatic increase in lipid peroxidation, proteolytic activity, protease gene expression, polygalactouronidase activity, nuclease activity, nuclear degradation, vacuolar autophagy, membrane leakage, disruption of cell membrane leading to cellular decompartmentalization, and above all, loss of tissue structure. The physiological consequences of physical and chemical changes in the membrane lipids include modifications in the membrane permeability, loss of membrane-bound enzymatic activity. The central senescence process seems to begin in the nucleus with the senescence of RNAs, which in turn are used to make certain proteins in the cytoplasm, ultimately resulting in the alteration of plasma membrane and loss of homeostatic ability. The active and rapid nuclear degradation is triggered by vacuolar rupture during programmed cell death. Senescence in plants is usually studied either as leaf senescence or flower senescence. But plant senescence can be best studied in flowers as it is the ideal tissue for studying senescence-associated events and changes. Flowers have been used as model systems to study senescence as the tissue is relatively homogeneous and chemical manipulation can be applied without substantial wounding. Now let's discuss about leaf senescence. Leaf senescence involves cell death that's controlled by age under the influence of other endogenous and environmental factors. Programmed cell death is a self-destructing cellular process triggered by external or internal factors and mediated through an active genetic program. The cell death occurring in leaf senescence is thus a type of PCD. Cell death in leaf senescence starts from mesophyll cells and then proceeds to other cell types. It also appears that cell death does not occur coherently but starts with local patches of early dying cells and then propagates to the whole leaf area. Leaf cells at the senescence stage show some distinctive structural and biochemical changes. A notable feature of cellular structural change during leaf senescence is the order of disintegration of intracellular organelles. The earliest structural changes occur in chloroplast, that's changes in the granular structure and content and formation of lipid droplet called plastoglobuli. In contrast, the nucleus and mitochondria that are essential for gene expression and energy production respectively remain intact until the last stage of senescence. This reflects that the leaf cells need to remain functional for progression of senescence until a late stage of senescence, possibly for effective mobilization of cellular materials. In the last stage of leaf senescence, typical symptoms of programmed cell death such as controlled vacuolar collapse, chromatin condensation, DNA laddering are detected in naturally sensing leaves from a variety of plants including Arabidopsis and tobacco. These observations imply that leaf senescence involves cellular events that ultimately lead to programmed cell death. Eventually, visible disintegration of the plasma membrane and vacuolar membranes appears. The loss of integrity of plasma membrane then leads to disruption of cellular homeostasis, ending the life of cell in sensing leaves. The cellular biochemical changes in the sensing leaves are first accompanied by reduced anabolism. The overall cellular content of polysomes and ribosomes decreases fairly early, reflecting a decrease in protein synthesis. This occurs concomitantly with reduced synthesis of RNAs and tRNAs. Further, cellular biochemical changes are most easily understood from the viewpoint of nutrient salvage. That's, for example, hydrolysis of macromolecules and subsequent mobilization, which requires operating a complex array of metabolic pathways. Chloroplast degeneration is accompanied by chlorophyll degradation and the progressive loss of proteins in the chloroplast, such as ribulose biphosphate carboxylase and chlorophyll A-B binding protein. Senescence-associated cysteine proteases, which are accumulated in the vacuole, also play a role in protein degradation. Lipid-degrading enzymes such as phospholipase D, phosphatidic acid phosphatase, lytic acyl hydrolase, and lipooxygenase appears to be involved in hydrolysis and metabolism of the membrane lipid in sensing leaves. The decrease in the RNA levels is accompanied by increasing activity of several RNases. Most of the fatty acids are either oxidized to provide energy for the senescence process or converted to a ketoglutarate via the glyoxate cycle. The alpha-ketoglutarate can be converted into phloem mobile sugars through glyconeogenesis or used to mobilize amino acids releasing during leaf protein degradation. After leaf senescence, we will discuss about the flower senescence. The onset of flower senescence is triggered by a number of internal, external, biochemical and molecular factors which initiates a series of physiological events orchestrated by plant growth regulators. Among plant growth regulators, ethylene is a clear regulator of petal senescence in some species such as carnation. However, in other species such as iris, hemerocallis, elastomeria, it appears to play a little or no part. Based on how the flowers respond to ethylene, two patterns of senescence with ethylene sensitive and insensitive systems are recognized. In ethylene insensitive systems, abscisic acid is thought to play the key role in senescence. The endogenous levels of abscisic acid and ethylene vary during flower development and senescence in the different flower organs. It is well established that ethylene is involved in the senescence of most of the flowers and coordinates senescence pathways and regulating floral abscission. It is generally believed that there is a gradual increase in the flower's sensitivity to ethylene with increasing age. In ethylene-sensitive flowers such as carnation, a rise in endogenous ethylene production triggers senescence and coordinates the expression of genes required for the process. In ethylene-independent flowers, senescence, as in iris, gladiolus, there is little ethylene production before or during senescence. Exogenous ethylene does not accelerate senescence, and ethylene inhibitors do not elongate floral longevity. However, some flowers, as in daffodil, intermediate pattern of senescence is exhibited. Without pollination, their senescence resembles that of ethylene insensitive flowers in that there is little ethylene production and only a limited response to inhibition of ethylene biosynthesis and action. However, pollination results in an ethylene-sensitive type of senescence with an associated rise in endogenous ethylene production, and as such application of exogenous ethylene accelerates senescence. In ethylene-insensitive flower systems, abscisic acid has been envisaged as being the key factor regulating flower senescence through an early and continuous accumulation in various flower tissues. Exogenous abscisic acid in daylily accelerated senescence-associated events such as loss of membrane permeability and lipid peroxidation. In narcissus, exogenous abscisic acid has been shown to lead to an premature accumulation of senescence-associated transcripts in tepals. Abscisic acid is thus thought to be primary hormonal regulator of flower senescence, and many of these senescence-related changes are brought about by it. These include ion leakage, change in lipid peroxidation, protease activity, and expression of novel DNases and RNases. It has been suggested that the signal for senescence is endogenous from the tepal cells themselves and may not require hormones as an intermediate signal. In Narcissus pseudo-narcissus, it has been shown that abscisic acid content increased in tepals of senescent flowers, which coincided with the appearance of visible signs of senescence, and exogenous application of abscisic acid has been found to enhance the premature accumulation of senescence-associated transcripts in the petals, indicating that abscisic acid induced the transcripts independent of ethylene. It has been reported that programmed cell death in very early stages of tulip petal senescence is triggered by signals other than ethylene, hydrogen peroxide or protease activity, and that the intracellular energy depletion is a primary early signal for programmed cell death in tulips. Using flow cytometry, it has been confirmed that nuclear fragmentation coincides with the death in petals after full flower opening. Recently, PCR-based subtractive hybridization has been successfully used in daffodil and iris to isolate large populations of genes associated with petal senescence. About 54 genes have been isolated from daffodil, including genes encoding a few regulatory proteins and several cysteine and serine proteases. It has been suggested that abscisic acid is a natural regulator of flower senescence and is involved in the regulation of senescence of ethylene-sensitive flowers. In rose and carnation, exogenously applied abscisic acid modulates an early increase in ethylene production, enhancing the sensitivity to ethylene. The flower longevity of ephemeral and long-lived hibiscus cultivars was found to be negatively correlated with ethylene evolution, and interestingly, abscisic acid also seems to be tightly linked with hibiscus flower life, showing the same trend as ethylene production. The endogenous concentration of abscisic acid in Hibiscus rosa sinensis during natural senescence was recently reported to be relatively low during early flower development, then peaked as the flower opens, and finally decreased during senescence. Abscisic acid may act in concert with or parallel to an ethylene signaling pathway during flower senescence. In plants such as rose, the expression of some genes encoding for ethylene receptors is induced under exogenous abscisic acid treatments. A large increase in abscisic acid levels has been observed in the gynoecium prior to or concomitant with the ethylene upsurge, demonstrated that ethylene perception is required for abscisic acid action in Hibiscus rosa sinensis, as treatment with silver thiosulfate, an ethylene antagonist, completely prevented the increase in abscisic acid levels. Thus, ethylene undoubtedly plays a major role in promotion of senescence, but role of other plant growth regulators cannot be ruled out. Cytokinins have been found to delay floral senescence in various plants like dianths, petunia, and roses. Cytokinins are natural antisenescence factors, and that their declining levels serve as trigger for increased ethylene production. Feeding carnation flowers with 6-methylpurine, an inhibitor of cytokinin oxidase or dehydrogenase, has been shown to increase lifespan of petals, suggesting that ethylene promotes inactivation of cytokinins and facilitates the senescence process. The role of auxins in regulating flower senescence is not clearly understood till date. However, auxins have been found to reduce ethylene sensitivity in some tissues, while in others they promote senescence by inducing ethylene production. Application of auxins to flowers has been found to stimulate senescence by hastening the rise in ethylene production. Gibberellic acid is known to delay senescence in some cut carnation flowers by acting as an antagonist to ethylene. In Grevillea, higher levels of gibberellic acid was found to enhance flower abscission rather than senescence, but the treatment did not enhance the longevity of inflorescences. Polyamines, which are ubiquitous in living organisms, have been implicated in a wide range of biological processes, including plant growth, development, and response to stress. The major polyamines comprises putrescine, supermidine, and superamine, which either occur naturally or as free bases or bound to phenolics or other low molecular weight compounds or macromolecules. Polyamines and ethylene use a common precursor SAM for their biosynthesis, but show opposite effects in relation to senescence. Polyamines have been reported as an effective anti-senescence agents and have been found to retard chlorophyll loss, membrane deterioration, and increase in RNase, as also protease activities, all of which help to slow the senescence process. Abiotic citruses also have a bearing on the regulation of senescence. Petal senescence is hastened by wounding of the flower parts. This damage results in the evolution of ethylene, which may then induce degradative events. The wounding effect is mimicked by ethylene application to unwounded flowers and is eliminated by adding narbornadiene, the inhibitor of ethylene action to wounded flowers. Drought citrus has been shown to shorten the life of many flowers, such as carnation, by increasing the abscisic acid level. Senescence in the modular sense is generally considered a beneficial process which increases the individual fitness by allowing a plant to get rid of the old reductant structures or as prelude to the onset of harsh environments. But in evolutionary sense, senescence is a deleterious phenomenon which decreases the fitness of the individual. Leaf senescence plays a major role in the survival of several species. Drought-induced leaf senescence contributes to nutrient remobilization during citrus, thus allowing the rest of the plant to benefit from the nutrient accumulated during the lifespan of the leaf. In addition, drought-induced leaf senescence, especially when accompanied by leaf abscission, avoids large losses through transpiration, thus contributing to the maintenance of a favorable water balance of the whole plant. Drought-induced leaf senescence occurs gradually and is characterized by specific macroscopic, cellular, biochemical, and molecular changes. Senescence is a correlative event and is the cumulative effect of the formation of flowers, fruits, seeds, apiaces, and roots. Senescence is a mechanism which works in the favor of maintaining an ecological balance both for the internal milieu and external environment of a species. Selective and temporal removal of flowers is species specific and carefully tailored to its ecological requirements. This is important firstly because the flowers can be substantial sink on the plant resources and as such energetically expensive to maintain beyond its useful life. In addition, its architecture has been exploited by pathogens that use the stigma as a point of entry and thus the flower poses an added risk of pathogen attack. Thus, regulated flower senescence is a beneficial process for the plant. Keeping in view the factors mentioned earlier, it seems that senescence is a deleterious phenomenon which pay heavily on the plant's life. But at the same time, it's important for the survival of plant under stressful conditions and at the peak of completion of the life cycle. To conclude, let me highlight some of the main points of today's lecture. We have so far understood that senescence is the final event in the life of many plant tissues and is a highly regulated process that involves structural, biochemical, molecular changes that in many cases bear the hallmark of programmed cell death. We have also learned about the main changes occurring in flowers and leaves during senescence. It is pertinent to mention that senescence can be considered as a necessary evil as it helps the plant to cope with the unfavorable conditions despite paying heavily on the plant resources. Thank you very much.